This video is going to be about DNA. And yeah, so go find any DNA questions. Give you a second. Okay, so the genetic code, it's the order of the bases of DNA. So the order of the bases of DNA is called the genetic code. And then the definition of a codon. A codon is a triplet of bases that code for a specific amino acid. And then amino acids are joined together by peptide bonds to form a polypeptide chain. Then what is the definition of a gene? A gene is a sequence of bases on a DNA molecule. Not all genomes code for proteins. And then introns are the non-coding sections of DNA, and exons are the coding regions. So semi-conservative definition. So for semi-conservative replication, so firstly, it's a type of DNA replication where a new strand is formed and an old strand is conserved. So what does it mean by overlapping? By non-overlapping, sorry. Non-overlapping means that triplets are only read once. The triplets do not share bases. What is the definition of degenerate? Degenerate means that more than one triplet codes for the same amino acids. It reduces the phenotypic triplet code for the same amino acids. For example, like mistakes in the bases, like base deletion, insertion, substitution. What are the effects of mutations? It can cause cystic fibrosis, sickle cell anemia, and production of sticky mucuses. Okay, this is the process of DNA replication. It's done in 4 to 5 parts, so that is altogether 4 plus 2 plus 5, 11 points that you should have. So first, the double helix unwinds. Second, hydrogen bonds break base pairs. Hydrogen bonds break between complementary base pairing. 3. Using enzyme DNA helicase. And 4. It separates into 2 strands. Then both strands are used as templates. Complementary base pairing occurs between 3 nucleotides and the template strand. The 5 points at the end. Number 1, adjacent nucleotides join. Number 2, biphosphodiester bonds. Number 3, formed in a condensation reaction. Number 4, using enzyme DNA polymerase. And number 5, hydrogen bonds form between bases on original and new strands. Now we have the process of transcription in the points 4, 3, 5. So altogether you should have 13.7 plus 5, 12 points. So number 1, a molecule of mRNA is made in the nucleus. Number 2, hydrogen bonds break between the complementary base pairs. Number 3, the DNA uncoils. Number 4, separating the 2 strands. Number 5, one of the DNA strands is used as a template by RNA polymerase to make the mRNA molecule. Number 6. The DNA template is called the antisense strand. Number 7. 3 nucleotides line up by complementary base pairing. Number 8. Adjacent nucleotides are joined by phosphodiester bonds. Number 9. Made by RNA polymerase. Number 10. Thus forming a single-stranded molecule of mRNA. Number 11. mRNA then moves out of the nucleus through a pore. And the last point, number 12, it attaches to a ribosome in the cytoplasm. The next process is translation, which is 3 points. So mRNA attaches to the ribosome. That's the first point. tRNA collects amino acids from the cytoplasm. Point 3, and carries them to the ribosome. Point 4, tRNA is a single-stranded molecule with a binding site at one end. Number 5, so it can only carry one type of amino acid. Number 6, and a triplet of bases at the other. So on one end it carries an amino acid, and the other end a triplet of bases. Number 7, tRNA attaches itself to mRNA by complementary base pairing, which means that 2 molecules attach to mRNA at a time. Number 7, amino acids attached to RNA molecules and joined by a peptide bond. Number 8, this process is repeated, thus leading to the formation of a polypeptide chain. Number 9, until a stop codon is reached on mRNA and ends the process of protein synthesis. Oh, the start codon is AUG, which is methionine, and the stop codons are either UAG, UAA, or UGA.